Okay, so let's discuss about the structure or the clover leaf. It is the structure of tRNA, or we can say that it is the clover leaf model. Okay, so as I told you in my previous video that this one model is proposed by Hawley in 1965. Okay, then he also told us that each oh sorry each each tRNA uh, contains the molecular weight as 25,000 AMU, and each uh, each tRNA consists of 70 to 90 nucleotides in it. Okay, then it is the smallest size tRNA is the smallest molecule. I told this in very detail in my previous video. So let's move towards the structure of the tRNA. Move towards the model or the structure of the tRNA. Okay, so this one is the structure of the tRNA. So let me give you the introduction of this one. So this one, the upper one area is known as the acceptor arm. This one, the area or the arm on the right hand is known as the T loop. Then this one lower side area is known as anticodon loop, and this one is known as D loop. Let me tell you that why it is known as acceptor arm. It is known as acceptor arm because it will accept the information of amino acid from the triplet of mRNA, and it is known as the T loop because it will synthesize it will give the ribosome recognition. It will recognize the ribosome, and it is known as the D loop because it will synthesize the amino acid, or it will recognize the amino acid. It will store, these two loops will be storing the amino acid and ribosomes. From T loop, a protein will be synthesizing, and from over here the amino acid will be storing. And in anticodon, the information of anticodon will be leaving. These three are the anticodon, and for example, this one line is the mRNA 5 prime 3 prime, and these three are the information that is known as codon. Then when let me tell you, then as I tell you that this is the mRNA, so on mRNA there's different type of codons, as we are having A, U and G. These are the adenine, uracil and guanine, and there are different type of codons, nitrogenous bases are present that form a triplet. It is a triplet. Okay, up in codon there are several type of nitrogenous bases are present that form a triplet. It is a triplet. These are forming by the nitrogenous bases. These three are the nitrogenous bases. Then each triplet will code for an amino acid. As I told you that acceptor arm will accept the amino acid from the mRNA. So each triplet will create a code for a single amino acid, and that one amino acid get attached to the tRNA from the source of the acceptor arm. Then at 3 prime OH group is present, hydroxyl group is present. Why hydroxyl group is present at 3 prime? Because it will bind the amino acid with the strands of the tRNA. Then according to this model, there are 5 arms are present. Now let me tell you, this one acceptor arm is the first one, T arm is the second one, anticodon arm is the third one, and D arm is the fourth one, and variable, it is a vestigial part. It does not do any kind of important function in it. So this one variable arm is known as the fifth one arm. Then if we talk about the acceptor arm, so in acceptor arm there are 7 pair of nitrogenous bases are present and 4 single nitrogenous bases. For example, one nitrogenous base is present over here, one is here, one is here, one is here. इस तरीके से like this. There are several there are 7 nitrogenous bases pair that are present in acceptor arm and 4 single nitrogenous bases. If we will talk about one strand, so only 4 single nitrogenous bases are present in acceptor arm. Then in D loop 8 to 10 nucleotides are present in D loop. This one is the loop. These circles are the loops and this one part is the arm. Okay, this one is arm and these circle part, this circular region is known as the loop. So this loop contain 8 to 10 8 to 10 nucleotides are present in D loop. Okay, why it is known as D loop? D arm is known as dihydrouridine it is known as dihydrouridine. That does mean that it is known as D loop just because of this one. What is uridine? Uridine is when one ribose sugar will add with 2 uracil, so it will form uridine. It's a kind of a molecule. Then let me tell you that there's no loop is present at the acceptor arm. After that, let's move towards the anticodon arm. This one is the let me tell you that mRNA will bring the information that is known as codon to tRNA to acceptor arm. It will give the codon to acceptor arm, and from acceptor arm the information will synthesize. The amino acid part will get stored in this one, and the ribosomal part will stored in this one, and they synthesize with each other, and after that the codon will move towards the anticodon and anticodon arm, and the information will be getting stored in this one loop in the and that is known as anticodon, and this anticodon will move towards the ribosomal RNA and get attached to that one. Okay, G. So what is anticodon arm? And anticodon arm is made up of 5 nitrogenous bases pair. Okay, it is made up of 5 nitrogenous bases pair: uracil, adenine, guanine, thymine, cytosine, and uracil. Okay, adenine, guanine, cytosine, thymine, or uracil. These are the 5 nitrogenous bases that are present in the anticodon arm or the anticodon loop. In the anticodon arm. Then in this one loop there are 7 nucleotides. In loop of anticodon arm, 7 nucleotides are present. Then after that, among of these 7, 3 nucleotides will work as anticodon. These 3 will work as anticodon. As I told you over here that uracil, adenine, and cytosine will work are the anticodon. So these 3 will work as the anticodon, and the remaining 4 will be stored inside of the loop. Adenine, guanine, and thymine and thymine will be storing in this one loop. Then we are having T loop. So in T loop the in T arm 5 nitrogenous bases are present and 7 nucleotide are present in loop. There is a 7 and arm के अंदर 5 nitrogenous bases are present. Then what is the work of the T loop? It will synthesize the ribosome. The transport of amino acid is present in the cytoplasm to the site of the protein synthesis. We can say that it will do the ribosome recognition. Okay, T loop will do the ribosome recognition, and D loop will do the recognition of amino acid recognition. So this is all.